As mentioned today, I'm going to be talking to you about cryo-electron microscopy, and specifically single particle cryo-electron microscopy. So I guess just to kick us off today, I will be talking about single particle cryo-electron microscopy, but really from a beginner's perspective. So I'm hoping that lots of you in the audience maybe have never used cryo-electron microscopy before, and maybe don't know what it's about, don't know where to start in terms of thinking, well, is my project suitable? What kind of things might I be able to get out of this technique? So I'm going to really start basically today hoping to quickly guide you through the workflow from sample to structure, hopefully with lots of time for questions and discussions at the end. So when we talk about single particle cryo-electron microscopy, what we're typically talking about is a technique by which we take a macromolecular complex, for example, this virus captured here, and we take hundreds to thousands of raw images, each of which would contain tens to hundreds of raw particles. And then we apply averaging techniques to generate, first of all, two-dimensional class averages, and then eventually generate our hopefully high resolution three-dimensional structure. Essentially through iNEXT-Discovery, we can offer two kinds of access. One of those, which may be applicable to some of you in the audience today, is from 2D classes to structure. So if you're already in an institution which has electron microscopes, you have some experience with electron microscopy, then you might be in a position where you have grids, you've already prepared your sample, and you really just need access to a particular technology. We can absolutely help facilitate that, and we'd be really excited to receive applications from you. But today, and what I'm really excited that we can offer, is this sample to structure pipeline. So I just wanted to highlight before I go any further that through iNEXT-Discovery, everything that I'm going to talk about today, if you just have your sample where you've done some biochemical analysis and you think that you may be able to use cryo-electron microscopy to answer some of your research questions, but you've never done any kind of electron microscopy before in your life, iNEXT-Discovery offers you the opportunity to apply. At the moment, given travel restrictions, for you to ship and send your sample to us, and then we can work that up through the pipeline that I'm going to be talking through today, hopefully, again, all the way through to your structure at the other end. But I just wanted to highlight that to start off with, because I didn't want anybody in the audience thinking, oh, well, this is no good because I don't have access to a particular kind of technology or part of the workflow before you get into the cryo-electron microscope, because through iNEXT-Discovery, we can really support you through that whole pipeline, offering you not only access to equipment, but also the staff expertise in order to get there. So this is essentially what I'm going to be talking you through today. So first of all, is my project suitable? What we would typically expect before you would move to cryo-electron microscopy, a bit about sample preparation and optimization through to data collection and data processing. So kicking off, is my project suitable? Well, that really depends. So there's lots and lots of questions here, and I'm going to just run through a couple of key points. So the first one is electron microscopy the right method for the biological question that I want to answer? And we'll come back to this one a little bit as we go through. So really what I mean by that is, first of all, what resolution are you looking to achieve with single particle averaging? And everybody always says, oh, well, as good a resolution as I possibly can. Of course, because we're all many of us are structural biologists, and we want to achieve the best resolution possible. However, you don't necessarily always need a sub-3 angstrom resolution structure in order to be able to answer your biological question. If your question is simply where a domain sits within a wider complex, a 10 angstrom or 12 angstrom structure could actually be sufficient for you to be able to answer that biological question. Whereas if, for example, you're looking at small molecule binding, then you would need a much higher resolution to be able to achieve that. So it's always worth starting at the beginning and thinking, what resolution do you need to achieve? The second one is how big is your macromolecular complex? So routinely, electron microscopy or cryo-electron microscopy, single particle approaches have been traditionally applied to proteins with a macromolecular complex size of ordered density. Because remember, if your density is not ordered, then you won't be able to average that between individual particles, and therefore it doesn't count towards what we kind of talk about as that ordered mass. Typically, we're working with protein complexes 150 kilodaltons, 200 kilodaltons or larger. The smaller size limit, however, is decreasing all of the time, and structures have been solved for things like streptavidin, which obviously significantly sub 100 kilodaltons. But really, if we're getting below 120, 100 kilodaltons, the question is, how much order density is there? So if you've got something which is 100, 150, 200 kilodaltons plus, then electron microscopy may be an appropriate technique. And the really great thing is that we can work with complexes many, many megadaltons in size. So whereas, for example, with NMR or X-ray crystallography, you may start to struggle with analysis of complexes of that size. Typically, cryo-electron microscopy would be a tractable approach for those kinds of sizes of complex. The next element is homogeneity. So if we take our protein complex, how homogeneous is it when we're thinking about applying our averaging techniques? What are the dynamics of that specimen? If your protein is highly dynamic, then that may make single particle analysis more challenging, depending on the nature of those dynamics. Another question we always tend to ask are there existing related structures? Obviously, many of us are working on protein complexes where there are no existing structures, because that's typically the unknown thing. But if you are interested in a complex where there are existing structures, not necessarily even of the whole complex, but for example of subunits within that. So, for example, it's quite common for electron microscopists to be trying to determine the structure of a whole complex, but there are partial structures of individual parts of that complex solved by X-ray crystallography, for example. That can be useful information when thinking about how tractable your project will be to start off with. The next thing is buffer. So when you're working up your sample towards cryo-electron microscopy, there are certain things which we try not to include in the buffer, and those would include glycerol, high concentrations of sucrose, high concentrations of salt, and certain detergents. All of those things can either affect the formation of vitreous ice, which we'll talk about later, or the contrast that or basically the ability to see your particles with signal to noise. There are lots of, with a good signal to noise ratio. There are lots of really good reviews out there on sample preparation that I can point people towards at the end if anybody is interested in the specifics around that. And then finally, how much of your protein can you produce? What sort of volume and concentration? And again, typically analysis by electron microscopy requires orders of magnitude less material than structural analysis by X-ray or NMR, for example. So if you have a protein complex which is quite difficult to produce in large concentrations or volumes, then electron microscopy may be an appropriate technique for you to consider. So all of this comes back to, so we'll leave that there for a second. So preliminary work before you move to electron microscopy. Typically, if we were looking at an application with somebody saying, hey, I've got a protein complex and I'd like to move to electron microscopy, the kind of minimal expectation that we would normally have is a gel analysis, and depending on the complex, normally size exclusion chromatography traces. But of course, and there have been webinars earlier on in the series around this, there are a whole host of different biophysical techniques, biochemical techniques, which you may be able to use to characterize your complex before moving to electron microscopy. And typically, the more well characterized your sample is before entering the structure determination pipeline, the better your chances of success will be as you move later. I always like this quote: don't waste clean thinking on dirty proteins. So if you were accessing my facility through iNEXT-Discovery and you put in an application, you demonstrated that you have some gels and size exclusion traces showing a nice peak, and you shipped the protein to Leeds. Typically, the first thing that we would do when that sample arrived with is do negative stain electron microscopy. So in this technique, we basically take our macromolecular complex. At the bottom here, this is an amyloid fibril, and we embed it in something like, well, normally uranyl acetate, but uranyl salt of some description. That gives us this kind of image here, and making this grid literally takes a couple of minutes, to get it into the microscope. So you can very, very quickly assess for the purity, heterogeneity, aggregation, degradation of your samples. And if you spend a little bit more time collecting more images and then processing those, then you can also get some information about flexibility, stoichiometry of complexes, that kind of thing. So it's a really, really good starting QC method in our experience. So for example, if you have something that looks like this, where it's nice monodispersed particles, nice 2D class averages, that would be promising. If we have something more like this on the right, where we can see a lot more heterogeneity, it looks a lot more bitty, then that will be less promising for us to proceed, and we might decide to go back to the sample and take a look. So next stage, cryo-electron microscopy sample preparation. So when we're talking about cryo-EM sample prep, we basically take a grid such as this one here, which is just over 3 mil across. And then across this metal grid, you have these windows, which we call our grid squares. And suspended across these grid squares, we'll have a support film. Now, typically, the support film is made of amorphous carbon, and it can either be an irregular lacy pattern, such as this one on the left, or a regular pattern on the right, which would include something like quantifoil or C-flat supports. And in our cryo-EM sample preparation, the aim is for us to get a thin, and by thin I mean tens of nanometers, probably up to just over 100 nanometers, layer thick of our buffer, containing our macromolecular protein or macromolecular complex of interest in a whole host of different orientations. And then we would freeze that. And we freeze it using a plunge freezing device. So these are just two examples of commercially available plunge freezing devices. And I'm just going to show you a quick video, which I hope is going to work over the magic of the internet, but we'll see. So what we have is basically a bucket, which is full of liquids. Just going to pause this a sec. What we have is a bucket, which is filled, I've realised it's probably not playing very well for you, which is filled with liquid nitrogen, and inside here we have a cryogen cup, which is filled with liquid ethane. And what we're going to do is take our electron microscopy grid, which is on these tweezers here, and then that is going to get raised up into this chamber, which is humidity and temperature controlled. And then through one of the ports in the sides, we just use a pipette to apply small volume, typically 3 microliters of liquid, onto our electron microscopy grid. And then we use filter paper to blot most of that liquid away, forming a thin film, and the grid is then frozen by plunging into liquid ethane. Once it's in liquid ethane and the sample has become vitrified, we can transfer it from the liquid ethane, which is about minus 170 degrees C, into the liquid nitrogen, and then we can store that grid essentially indefinitely until we decide to image it. So this plunge freezing approach to basically fixing our specimen in a layer of vitreous ice is incredibly flexible. So we can use plunge freezing to freeze a whole range of different biological specimens, from whole eukaryotic cells, which are grown on grids, through bacterial cells, liposomes, viruses, macromolecular complexes. And really what we're doing here is freezing our specimen so quickly that rather than what normally happens when you freeze, which is that your water molecules rearrange to form ice crystals, we're freezing our water so quickly that it forms amorphous ice or vitreous ice, and essentially suspending our macromolecular complex in as native-like way as we currently know how. So that we can then image it in our transmission electron microscope. So after we've done that, what we want to do is get it into a microscope and have a look. And the first test is really, is your ice vitreous or not? And on the left here is an example of non-vitreous ice. We can see this black patterning here, but normally this is quite an unusual situation, and normally the creation of vitreous ice with your particles embedded in it is routinely achievable. Unfortunately, what's not routinely achievable is your proteins forming, as shown in A here, a nice monodisperse arrangement of particles in different orientations. And a little bit of a survey with projects that we have going through the ABSL facility basically showed that even for things we would consider to be fairly routine, such as icosahedral viruses, almost every specimen that we've had come through has required some kind of optimization in terms of the cryo-EM grids. And this basically means making some grids, taking a look, working out what's wrong with it, and then taking an educated guess, going back and making more grids. And typically, you know, these are the kinds of things which we often see happening. Sometimes your ice thins in the center and that physically excludes your particles, pushes them to the edge. Sometimes your particles form a preferred orientation. We'll discuss why that's important later, but basically it means that you can't get a decent average because you're only looking at the macromolecular complex from one angle. Sometimes it won't partition to the holes, which is where we want to image our structures. And the annoying and good thing is that there are lots and lots of different variables, and almost every protein complex that we work with behaves in a slightly different way on the cryo-EM grid. And that is frustrating, but the good news is that there are lots of different variables that we can then go back and adjust and change, and in order to get a cryo-EM grid which you can proceed to data analysis with. And that's the advantage of accessing cryo-electron microscopy through something like iNEXT-Discovery, the sample to structure route, because that basically means that not only will you have instrument access, but you will also have an expert staff scientist working with you who's worked through lots of different projects and generally has a good idea of the kinds of variables that are worth altering in the first instance to try and get you to this well-distributed position at the top. So, for example, here, this is an image where the ice is thinned in the middle and these virus particles are packed up against the edge. This is an example of preferred orientation. So for those of you with keen eyes, you'll see basically all of these little beta gals. They basically look like the same shape because they've got a preferred orientation. And, you know, we can do different things to tackle that. Sometimes there are just too fewer particles, and there are a whole range of different grid technologies and support films that you can use to tackle this. Another variable is the properties of the support film itself in terms of what treatment you give it. So how hydrophilic is it? What is the charge on that grid? That can also affect particle distribution. So for cryo-EM grid screening, for many projects and for many institutions, this is a real bottleneck. But through using a transmission electron microscope with an autoloader associated with it, so this is basically a mechanism by which rather than loading grids one at a time, you can load 10, 11 grids at a time into the microscope. You can screen grids in a more medium throughput manner. And I'm going to come back to other ways that we can make screening more efficient a little bit later when I talk about image processing. So once you've got your grid and it's got a good particle distribution and you're ready to proceed to data collection, then this is where you can, if you had that grid already, you could then apply from 2D structure to 2D classes to structure routes. And yeah, we can proceed to data collection. So I'm just going to show a time lapse here because I really like this. This is back from 2016 now, where we had our 2 Titan Cryos microscopes installed at the University of Leeds. So the facility space in Leeds, if you ever get a chance to visit, is, and although I'm biased, it's really very cool. So there are 2 Titan Cryoses located in what is an old television studio. So just on the north of that image, you can see essentially the director's deck up there, which is where all of the director's studio used to be. And that's where you sit up and control the microscopes. I should say we also offer control of the microscopes completely remotely from wherever you are in the world. Yeah, so we now have these 2 Titan Cryos microscopes in this studio space. And I should say that our facility, we're currently under tender to upgrade these microscopes. We're going to have £1.7 million of upgrades coming to our facility, Q1, beginning of Q2 next year, which is very exciting. So basically, once you've got to the stage that you've got your grid, you're going to come to a facility and collect, in our case, data with us on our Titan Cryos microscopes. And we do that by using a program called EPU, which stands for E pluribus unum. Excuse my Latin, not very good, but essentially that translates as out of many one, which I think is very poetic, because what we're basically doing in cryo-electron microscopy single particle averaging is taking many particles and using averaging approaches to turn that into one structure. So when you first come to collect data, there can be many different variables that you have to consider, and especially when users and researchers come to visit us for the first time, sometimes that can be a little bit overwhelming because there's so many questions. Which detector, what electron dose should I use, how many images do I need, how many images do I want, what magnification should I use, how many acquisitions per hole should I use a phase plate? And this is where I always say don't panic, because this is what facility staff are here for. So what we'll typically do is meet with you before your data collection session and run you through all of these kinds of questions and just talk you through all of the different options using the experience that we've had with a range of different projects to design a single particle data collection experiment that suits you. And then on the day, what we basically do is we take a low magnification atlas of the grid, and we'll use that to identify the Goldilocks zone, as I always call it, in terms of your right ice thickness. So at the bottom here, you can see the grid squares are basically too dry. At the top, they're too thick. And in the middle, we have this zone that is just right. So we can then go into each of those grid squares and select which holes we want to go and visit. And then within each one of those holes, you can put a number of acquisitions. So for example, here we have 3 different acquisitions around this hole, and off to one side, an area where we can perform autofocus and things like that. And typically that will take 4 to 6 hours to set up, and then you can press go and collect data from between 1 and 3 days, depending on your experimental setup. Normally 1 to 2 days is standard. Let's skip that. And then finally, I just want to talk very briefly, because I'm running out of time, on data processing. So there are a huge number of resources out there which talk you through the data processing pipeline. Out here, I've outlined the key steps, but essentially there are a huge number of programs out there now which are designed to walk you from your initial data right the way through to the creation of your EM density map. Which will look something like this at the end. I just wanted to highlight a couple of parts of this process. And the first one is what we call on-the-fly assessment of your data. And basically, the majority of facilities, certainly if you come to visit us, will do some kind of on-the-fly processing of your data. And what this basically means is that as a minimum, you will have motion correction and CTF estimation of your data, and then some kind of graphical output of what that looks like. And here, for example, you can see if we have a look at this data set, then the resolution seen in each of the micrographs is incredibly spread, and the astigmatism is all over the place. And this is clearly a very bad data set. So actually back in 2016 now, so I can't remember what was wrong with it, but clearly something very bad has happened. And then this would be an example of a slightly better data set, but where there still might be something going on we might want to investigate. For example, here we seem to have a bit of a double distribution. But anyway, the majority of these micrographs are showing good resolution retained in the images. And what this basically does, it gives you the ability to quickly look at what's going on and just check to see that some of the parameters, so for example, motion is within the expected range. And it gives you an opportunity to say, hey, is there something going on here that we need to go and take a look at? And I mentioned earlier about making screening more efficient. So one of the things that we're particularly excited about is the prospect of starting to do screening in a slightly more intelligent way. So at the moment, typically what we do is we put a grid into the microscope, and we'll manually go and take 10 to 20 images if the sample looks good, and then manually inspect those and go, yes, that looks okay, let's proceed to data collection. However, what we're starting to do more and more of now, and this work was done initially by Matty Danza, who's now gone to work for CCP-EM, is to collect a small, say half an hour, 45-minute data collection, and then run it through a completely automated pipeline to then generate a three-dimensional reconstruction. And this can quickly help you identify, for example, situations where you might have a preferred orientation, something like that. And these are the kinds of pipelines which we're hoping to be routinely implementing for all users, so both external and our internal users. So I just wanted to finish off by really highlighting that cryo-electron microscopy, because we can deal with proteins across quite different size ranges, because we can deal with relatively low volumes and low concentrations of the specimens. It can really be used as a structure determination technique for a whole wide range of different proteins and macromolecular complexes. And here's just an example of some of the structures which have come out of the facility in the last year. You can see we've got a real range here from really quite labile and awkward macromolecular complexes, amyloid fibrils, membrane proteins. This is structure just out in nature, and this was again very, very fiddly macromolecular complex. So we're able to use single particle pipelines now to tackle ever more challenging macromolecular complexes. So I think I'd really encourage you to use this as a technique and consider whether it might be a workflow that is useful for you. So as I mentioned at the beginning, we offer access through iNEXT-Discovery. Check us out on the website, but you can always email us at cryo-EM@leeds.ac.uk if you want more information. And as my very last slide, I just wanted to announce that early in the new year, we're going to be launching our Wellcome and MRC co-funded community-based training program in cryo-electron microscopy. So this program was particularly set up to try and build skills within the international community and give people who don't necessarily have access at their home institution to these techniques and technologies the ability to get hands-on training. So if you're interested not only in applying for time and accessing for your project that way, but if you want to get real hands-on training, then watch the space, and there'll be announcements coming out on CCPM, 3DEM, Twitter, all the usual channels. Okay, that's it. I said a lot of words. I'm going to stop talking. Thank you very much.